And and so this is natural recovery basically from logging and acidification that's created this gradient in this lake. So the real question after we published this paper that fish are nothing more than tree leaves was basically how general are these patterns? Because we've just looked at one lake here. So we got funding from the Natural Environment Research Council to bring some of the real leading experts on stable isotopes and aquatic food webs to Cambridge to form what we call the Lochtony Working Group. And we brought together nearly 600 observations from 150 lakes worldwide. So this is just where the different lakes were located. So the observations that we collated span the boreal to the more like warm temperate part of the US. And what we discovered was that half of all zooplankton, so these are the number of observations in our data set, half of them were comprised of at least 42% terrestrial material. So half of all zooplankton are made up almost half of terrestrial material, but there is a lot of variation here in this plot that you can see. And so we found that by relating this variation in the terrestrial resource use by zooplankton to surrounding land use and in-lake characteristics, that the terrestrial support actually turned out to be greatest, and it kind of makes good sense, in the lakes with really strong physical and hydrological connections to the surrounding catchments that were rich in above and below ground carbon. One of the things that I guess we didn't see in the video, but I glossed over, was that what's really key to link this exchange between land and water is what happens at the base of the food web. So the first step in that process in the video would have been the microbes chewing away at the leaves. So the traditional model of aquatic food web ecology for a long time was that basically by using sunlight and dissolved inorganic carbon, phytoplankton fixed carbon, and that's transferred upwards through the food web into zooplankton and into fish. And in addition to that, there's something called the microbial loop, especially the viral shunt. We'll get to viruses soon. And what this involves is basically the microbes are recycling dead biomass from fish and zooplankton, respiring that out as dissolved or inorganic carbon, and that gets refixed by phytoplankton. So for a long time, this was the traditional model of how aquatic food webs worked. Now, in recent years, discoveries by our group and others have taken basically these food webs and linked them back to land, to particulate organic carbon and terrestrial dissolved organic carbon. So what we did in my group was to really test how this energy transfer from land into water varied with these sources here, with these terrestrial sources. Now we know that these terrestrial sources are changing in at least two major ways. So the first is really that there's a strong trend in greening of the landscape measured as this leaf area index from remote sensing imagery. So all we have to take away from this map is that places are getting greener because of climate change. And that's predicted, here's the change in LAI into the future, that's predicted to carry forward, especially at these high latitude Northern Hemisphere sites. So forests are getting greener. That's all you need to take away from this. Likewise, there are clear predicted shifts in the dominance of different tree species. So this is a great map because you can tell an American made it, because everything up here is just a great white void. Nothing exists north of the US border. But what this does show is that in the US, forests here in the Northeast are predicted to go from this maple, beech, birch dominated forest to sort of more oak dominated forest. So basically the other thing that's happening is that the forests are greening and they're also changing the type of greenness that's coming into them. And so what we did in my group was to carry out this basically climate change experiment to simulate what might be the effects of changes in forest composition and changes in forest biomass on the linkages between land and nearshore aquatic ecosystems where those terrestrial inputs are deposited. And so we set up an experiment. It's the related experiment. It stands for Restoring Ecosystems by Linking Aquatic and Terrestrial Ecological Dynamics. And what we did was we simulated these changes in these mesocosms that we deployed in the nearshore where organic matter accumulates. So here's just a photo of these mesocosms. They're basically bussing bins from like a restaurant, probably about this big, and different quantities and different types of organic matter that we supplied them with. And the really important part here is that there's this little thing sticking out here, and that's a pore water sampler. So that just sat, that's a syringe with a mesh out with some pantyhose on it. And it just sat right beneath the sediment, allowing us to sample the water continuously. And then we replicated this experiment in 3 lakes in central Ontario with different overlying water qualities. So the differences in the lakes tried to capture 2 additional drivers of global change, namely nutrients and light. So, oh, because I'm touching it, it's a smart screen, isn't it? I should stop touching it. Okay. So, so we had lakes with low nutrients, but changing light levels, okay, associated basically with this idea of browning of the water. And then we had a low light, high nutrient lake. And we didn't have this 4th corner here of sort of high nutrients, high light, because that's not actually readily available. And then I have another video, but this one doesn't have any audio, that's okay. And so what you can see here is that we deployed this experiment in the near shore where terrestrial organic matter accumulates. And we asked how those microbes, those gatekeepers at the base of the aquatic food web, were changing in composition and function with the different terrestrial organic matter inputs and the consequences for carbon cycling. So you can see here, this tubing, that actually runs down into those syringes that I told you about. And then the tubing comes out onto the surface. You're gonna see that in a moment. There we go. So that meant we could just basically pull up water constantly. And so we did this every 2 weeks for 3 years. And this involved a lot of people, some of which are shown here. Our biggest year being in 2016, in fact, and it felt at times like my job was to run a summer camp for young adults. And yeah, so one of the major things we discovered with all of this was that different organic matter treatments shifted how the microbes function. So this is quite complicated, but we'll go through it, okay? So this is work done by Chloe Orlon, now at UC Riverside, as part of her PhD in Cambridge. And we have a heat map, okay, of the log fold change in abundance of a whole bunch of functional genes in our sediments. So from nearly 3,000 genes, we plotted the functions with the strongest changes in abundance over a 1-year period, ordered from the largest increase to the largest decrease. That's all you have to know. Don't try and read anything on here. Now, what I want you to take away from this is that the 2nd and the 3rd largest increases were associated with the intensification of oxidoreductase activity. So these are extracellular enzymes involved in the degradation of organic matter, and methanogenesis, which of course is a byproduct of microbial decomposition and is particularly concerning because methane has 25 times the warming effect as carbon dioxide. Now, these changes were kind of pretty consistent across the treatments, though there were some small treatment effects. And as part of a master's, Amelia Fitch, who is now at Dartmouth completing a PhD, found that as the organic matter became more complex, more aromatic, so larger values of the specific UV absorbance or SUVA, in part because of more of a terrestrial input, then bacterial production actually decreased. And as we show in this paper, it's likely because the bacteria were investing in the production of those oxidase and hydrolase enzymes to access organic matter, and that actually came at a metabolic cost. So in the dark lake only, what we found was that bacteria respired more carbon in those high SUVA conditions where they were also less productive. And we think that this difference arose because there were fewer low molecular weight compounds such as from photooxidation in the dark lake. So bacteria had to produce those costly enzymes to access carbon, which caused them to incur this metabolic cost. And so you can think of it maybe like eating, I don't know, maple syrup, right? You're going to be really quick to absorb the sugar, but it might, and it's going to be a lot easier to digest than something like celery that's going to be really rich in lignin. Now, the other reason we might have not seen a response here in the Clear Lake is that we found that the microbial communities that colonized the mesocosms had much lower abundances of these enzymatic genes for organic matter degradation. And so what we've done here in this plot is we've taken, so each point here is one of those mesocosms I showed you, and they've been identically treated in the clear and in the dark lakes. And all we've done is compared the abundance of these relevant functional genes. So if everything was the same between the clear and the dark lake, all the points would be sitting on this one-to-one line, okay? And you can see quite obviously that's not the case. All the points are sort of beneath it in this corner here, where what it's telling us is that there are much higher abundances of these functional genes in that dark lake. And so in the clear lake, there may be no selection either from evolution or the environment for individuals to have these enzymes, as they're more easily accessible sources of carbon available. And we find a very similar story in terms of methane concentrations that we published last year in ISME in a paper led by another former master's student, Kurt Yakimovich. Okay, so these are in the mesocosms. What's going on in nature? Well, we've tried to validate these findings, and we went back to that really nice lake in Ontario that I told you about, and we sampled sediments in the deltas beneath each one of these catchments that varied so much in surrounding forest cover. So this is a horrible looking statistical model, a path analysis model. And it's kind of like a map, okay? So it's showing the predictors. So these are where the arrows originate of different response variables. So that's where the arrows end. And the numbers on here are just the associated proportion of variance that one thing explains onto another. Now, the key thing I want you to take away from this is that the genes encoding oxidase enzymes, okay, which allowed the microbial community to access organic matter and increase carbon dioxide production in the natural lake. These genes became more abundant as there was more terrestrial carbon in the sediment. Again, this likely arose because there were changes in the relative abundance of microbial communities rather than changes in the presence or absence of specific taxa. And in support of this idea, we found that communities sampled at this end of the lake weren't actually very different from those sampled up here. So there wasn't a lot of variation in the composition geographically. So sediments beneath very different catchments at either end of the lake had nearly identical composition. So this idea that everything was actually everywhere. Rather, it was the communities were varying primarily because of the biogeochemical conditions in the sediment, like the percent of terrestrial carbon. And this result actually mirrors what we're seeing in other systems. So this is, we were just talking about. With collaborators at the University of Alberta, we've been measuring how microbial composition changes along a really large space for time gradient of future permafrost thaw in the Northwest Territories. So this is, this is too small, you're not going to see that. So what we've been doing is relating greenhouse gas fluxes in these peatland lakes to the microbial communities. So what's really great, like I was just talking to Guilherme about, is that in Alberta and the Northwest Territories, if you want to get up into permafrost, you don't have to fly $2,500 from Montreal to Nunavut or wherever. You just get in a truck and drive. And so here, our collaborators have a 1,200 kilometer transect that spans basically all the different permafrost zones, from sporadic all the way to continuous. And in each one of these areas, we have several lakes, peatland lakes, that are experiencing permafrost thaw that we've been able to study. And work led by Johanna Winder in our group found that the activity rather than the taxonomic composition of microbes, again, changes along this gradient. And here are some of those data that we've contributed to a really amazing paper led by Kenzie Kuhn at Alberta. And in this paper, what Johanna found was that the markers of methanogenesis and methanotrophy increased as permafrost was lost. So as we went from lakes in the continuous to sporadic to no permafrost zones. And there's a more detailed paper on these changes coming soon. And these findings are kind of consistent with another paper we published last year in Global Change Biology that found that the loss of permafrost isn't really changing the food web in streams, in these peatland streams. And you can ask me more about that afterwards if you're interested. And so we're now trying to shift some of this work in my group, at least, kind of more locally to James Bay and northern Quebec. So if anyone works there, it'd be great to talk about that afterwards. So the last point, I guess, I want to mention about all of this. How are we doing on time? Probably going very fast. So apologies. Like I said, I have not done this in a while in person. So the last thing that I want to talk about is what really it turns out really controls microbial dynamics. And if maybe you can guess it, it's viruses. So Lucas Braga, a postdoc in the group, worked with that related experiment that I just finished telling you about. And what he discovered was that the composition, the molecular composition of carbon in these lakes actually shifted towards low molecular weight compounds that create clearer waters as there were more viruses. And so this plot here is just a network diagram that shows the connections between over 150 different, basically viral taxa that we discovered in our sediments, and their connection with different classes of carbon compounds. Okay, so where there are more, so take the lignin group here, for example, there might be 3,000 different types of lignin molecules in this group. So that's why there are multiple lines coming out from lignin. And then we've basically mapped all the statistically significant positive correlations between a molecule in any of these classes and the different viruses. So where you see thicker lines from the viruses, they're better connected to the microbial, to the organic matter composition. Okay. And what we discovered from this analysis was that viruses were disproportionately positively associated with these amino sugars. And these amino sugars are important. They're actually the main components of bacterial cell walls, and they're what's mainly released when cells die because they're killed by viruses. And although viruses killing bacteria would also allow these larger molecular weight compounds to accumulate like lignin, which is indeed what we found in the negative network. So you can see that the number of connections with lignin actually increases massively in these negative networks, okay? And we think there's lots of lignin when the viruses are absent and they're not killing the bacteria that break down these compounds. But despite that, this change in the amino sugars was several times larger and enough to actually kind of dilute this effect of the slowdown of microbial decomposition. So that then caused the water to become clearer as there were more viruses in addition to the viruses themselves killing bacteria and stopping decomposition. And so as just further evidence of this is an estimate here of the viral load in these samples, which we found both reduces bacterial production and extracellular enzyme activity. So this is phenol oxidase, a really critical enzyme used to degrade organic matter. So the key point here is that viruses have the potential to completely shift the energy transfer in these lakes by making the water clearer, killing bacteria, in addition to their direct impacts on carbon cycling and the amount and the types of compounds available for microbes to use. Now, this work is stuff we're actually trying to substantially expand. So Lucas has received the Marie Curie Fellowship to work on the biogeochemical consequences of ancient viruses in thawing permafrost soils, and maybe stuff we'll do in northern Quebec too. I don't know yet. We're just starting. And we're actually hiring a postdoc presently for a new project looking at viruses in watersheds of Nunavut with Inuit partners in Kuujjuaq. If anyone is interested in talking to me later about that, the job's up for another 2-3 weeks. Okay, so really the take-home messages from this work are that, and I keep standing in front of everything. So future increases in terrestrial organic matter have the potential to reduce energy transfer through the food web by accelerating microbial decomposition. Okay, but the extent to which that happens depends on local environments and the adaptations of the microbes that live there. So in other words, you can feed microbes the exact same meal. We've done that, right? We've literally given them the exact same thing in different lakes, but their response depends on the local conditions, such as light or pre-existing carbon sources, at least in the very short term. Okay, so all this suggests that if we can make predictions about how terrestrial inputs are changing, we might be able to scale our results to the wider landscape using solely environmental data. And to just give you an example of that, this is work that Eric Emelson did as part of a postdoc. He's now a research scientist at the Canadian Forest Service. And what he discovered using a really scaled down version here of our mesocosms was that methane production was 400 times higher from sediments composed of macrophyte leaves, Typha latifolia, the cattail, compared to coniferous or deciduous litter. So this is a log scale, okay? These are really massive differences between the Typha and the other two. And it turns out that the coniferous and the deciduous litter, actually, the methane production was no different from the control treatment where we didn't add any organic matter. And it turns out that this result is because the forest litter is actually really enriched in these polyphenolic compounds, which can suppress the activity of methanogens and hold in place large quantities of carbon in sediments. So this was pretty much unknown before now. And the way we've reached that conclusion is that we've confirmed that there was inhibition. We actually added known methanogens to these different sediments and found the exact same results. And we also sequenced the abundance of the methanogens using genetic markers. And again, it sort of mirrored these results. So really 2 additional lines of evidence suggesting that the methanogens were being inhibited. And so what we've done is that from these results, we've taken species distribution models to forecast how cattail distributions will spread under a warmer climate under the Lake Ridge boreal shield that we've studied. And so these are the sort of current day projections of the where we've taken the occurrence of cattail and converted that into an estimate of the greenhouse gases that are going to be released from those lakes versus the projected estimates. And all you really need to see here is that there's going to be larger estimates in the future, and we actually quantify that as about a 70% increase in the greenhouse gases released from this region solely due to the spread of this one macrophyte species. So the cattail alone. But the spread of that one species isn't the only thing that's happening on the boreal shield. There's a lot of other processes. Disturbance is one that's actually quite pervasive. And work by Sam Woodman during his PhD has tracked how outbreaks of disturbance of insect defoliators actually impact the land water nutrient cycling. So it turns out that insect outbreaks, so maybe you can see this gray area here, this gray big chunk, a good sort of half of Ontario, insect outbreaks occur at least every 5 years across this area of Ontario. And so these are in different lake catchments since 1985, the number of outbreak events. And using 4 decades of water chemistry data from partners in 12 of these lakes, we've discovered that dissolved organic carbon concentrations actually decreased by about 30% after insect outbreaks. So what we have on this plot here is the dissolved organic carbon concentration over the growing season. The points are just the means and the errors, and the different colored lines here are the percent of the catchment that has been damaged by insects. So we have really good aerial flyover data where the insect outbreaks are mapped over these catchments. And in most cases, if there's an insect outbreak, 99% of the catchment is damaged. And so what we find is that there is no change in the dissolved organic carbon concentrations over the start of the summer, but when the caterpillars transition from the feeding stage to the adult, there's this decline in the carbon concentrations if you compare the green lines to the black lines, and that persists throughout the rest of the growing season. And it turns out that these effects were even stronger as more of those catchments were covered by deciduous trees. In terms of the inorganic nitrogen concentrations, these actually more than doubled because of the insects. So I don't know if you can see that down here. So again, no change early on in the season. And then by the late season, the black lines here is above that green line. So where there's a lot of insect damage, the nitrogen concentrations are like double in the water in the lakes. This kind of all makes good sense in a way, because what the caterpillars are doing is they're just removing carbon from the forest to assimilate it into their biomass. So there's less carbon available to enter from forest litter when leaves fall in the autumn, right? That's what we see, especially down here. This is what we've highlighted in gray here. This is the leaf fall period. And instead, they've taken this carbon and they've converted it into nitrogen-rich frass or poo that they just excrete onto the landscape, and that just gets flushed into the water. And so then you get this spike. As they're taking the carbon, they're removing it from the leaves, using it to grow, and then out the back end comes nitrogen-rich waste. That's what ends up in the landscape. So now one of the things you might be asking is, okay, well, that kind of sounds cool, but I mean, does it really matter? So like here, I'm showing you a change maybe of about 1 milligram per liter in dissolved organic carbon. Is that really relevant? And so what we've done is we've taken these estimates of the effects of insect outbreaks, and we've compared them to long-term trends reported in over 250 boreal and north temperate lakes over a similar sort of 25, 30-year period. And what we found was that a single year of these insect outbreaks, so that 1 milligram per liter effect, was basically large enough to offset 2 years of accumulation of dissolved organic carbon that was happening naturally in these lakes. In terms of nitrogen, the insects, one single outbreak could exceed intraannual changes in nitrogen by about 12 times. So these are actually quite important effects that the insects are driving on these landscapes, and something that, again, needs to really be thought about when we're trying to track what's happening to the land water carbon cycle. Okay, so we're doing okay. Okay, so for the next part of the talk, I want to just return to that food web I told you about. And so I'd argue that with the advent of genomics, we're really starting to understand the role of microbes at the base of the food web. But it's become increasingly apparent from our work that there's actually a lot of variation in their responses to terrestrial organic matter. And I'd actually argue that that's almost creates another sort of black box here in terms of the carbon. And historically, much of our understanding about this black box, about carbon, has come from quantifying organic matter as dissolved organic carbon. So we just measure DOC concentrations like I just showed you. But again, I'd argue that's almost like seeing through a keyhole. There's much more to organic matter than just carbon. So using ultra high resolution mass spectrometry approaches, what we're doing now is trying to sort of widen this keyhole to see hundreds of compounds scattered in a water sample. Now, these compounds can include lignin and cellulose and tannic acids, so carbon in all different forms and with all different functions. And this diversity of compounds, or what's called chemodiversity, so mirror the term biodiversity, is actually tremendous. So in those sediment experiment that I told you about, we find thousands of different unique molecular formulas in the natural environment. And if we survey the natural environment, we find actually like tens of thousands. So that's, to me, a lot more that is implied by a single element of carbon. We talk about carbon, but carbon is like 10,000 different things. And so now you might be saying, okay, well, that sounds kind of cool, but like, who cares? What does this diversity do anything? Is it all just carbon at the end of the day? Well, here I'd say that biodiversity science provides a really powerful framework for predicting how the functioning of ecosystems might actually vary with the number and abundances of the molecular formulas in dissolved organic matter, this chemodiversity. And so I'd say that like additions of species diversity to a local community, each unique molecular formula in a DOM mixture introduces new properties and functions. So, for example, the addition of labile aliphatic compounds can provide novel substrates that promote microbial coexistence, degradation, and assimilation, and then that's going to influence what these microbes do to key ecological processes, such as carbon cycling. And so ecosystem function and health can very much vary with chemodiversity, depending on its sources and composition. And this is something that people are just really starting to think about. And in 2019, we published the first paper that actually linked this ultra high resolution measurement of dissolved organic matter to ecosystem function. And so returning to two of those lakes in our sediment experiment in Ontario, we found a really strong connection between microbial and chemical diversity. So along the x-axis here, we have this estimate of the number of molecular formulas in one of those little mesocosms that I showed you, and a measure of the microbial diversity. Okay, quite a strong positive relationship. And here, this plot shows that as those chemicals became more similar, so too did the microbial community. So this is implying a really strong connection between the two. So each point here is basically the pairwise comparison between two mesocosms, and we've just calculated how similar those mesocosms are in their chemicals, how similar they are in their microbes, and a strong connection. And it turns out that this had really large consequences for ecosystem function. So here we're plotting the CO2 and methane concentrations in relation to chemodiversity, and I'm showing you what are called partial residual plots. So that's why these numbers look like that, and these just means it's a statistical technique where we've controlled for other reasons, at least in the visualization, for why the variables might vary. So like the dissolved organic carbon concentration and the experimental lake. And so we're just isolating in this plot for visualization purposes the effect of chemodiversity. And so what we found was that over the observed range of chemodiversity, carbon dioxide and methane concentrations increased by a mean of 16 and 55 times respectively. So big changes. And we further found that chemodiversity itself strongly varied with those experimental treatments I told you about earlier. So an increase in sediment organic matter from 5 to 25%, as can happen under from greater forest cover under a warmer climate, was enough to raise chemodiversity so that it increased the greenhouse gas concentrations. So when we add together CO2 and methane, by nearly 1.5 times. And we could make similar comparisons between that light and the dark lake to predict the effects of widespread browning or darkening of northern waters. And in that case, chemodiversity increased by 1.2 times in the darker lake, which was enough to increase the greenhouse gas concentrations by 2.7 times. So really compelling evidence, I would say, that chemodiversity is important. But like with our forests fuel fish growth paper, this was only a couple lakes, right? you might be wondering, how general is this pattern? So with funding from the European Research Council, what we've done is we've carried out the largest survey of chemodiversity in Europe's lakes, from Calabria all the way to Nordkapp in northern Norway, sampling over 100 lakes. And thank God it was done in 2019, because it would have been a nightmare. And so alongside chemodiversity, so that's what we have in this map here, we've measured ecosystem multifunctionality in each lake. And this is simply the sum of 5 functions related to carbon cycling. So methane, carbon dioxide emissions, and we measured a bunch of things related to ecosystem metabolism, like total respiration, maximum primary production, and bacterial protein production. So the team was really busy. So there was, it would have been quite nice to have done. So they spent 6 months driving across all of Europe. And in the evenings, they didn't even sleep. They were just running all these incubations right wherever they were in the van that they were living in. And so you can see here that maybe, maybe there appears to be these latitudinal trends. Maybe there's a bit more function in the south than in the north. Maybe a bit more chemodiversity in the north than in the south. It's quite hard to tell from these maps without considering other confounding variables. And so what Jeremy Fonvielle did, who led this work, was he led a path analysis. He fitted a path analysis to the data. And like I said before, this is kind of a map that shows inferred cause and effect relationships. And in this path analysis, the dashed lines just indicate negative associations, and the solid lines indicate positive associations. And so what we found was that there was more chemodiversity in the northern latitudes with less net primary productivity in the catchment. So a negative relationship between NPP and chemodiversity. And our index of multifunctionality actually increased with chemodiversity towards northern latitudes, but this was somewhat offset by the positive effect of warmer mean annual temperatures in the more southerly sites. So that's why the patterns on the maps wasn't as clear as it could have been. But the key point here is that chemodiversity is actually having quite a strong effect on ecosystem multifunctionality in nature. And this effect, we actually compared it to the effect of biodiversity on ecosystem function that's reported in the literature, and this effect was actually larger. So chemodiversity really matters for how lakes function. Now, another thing you might be wondering is, okay, well, how general are these patterns throughout the year? Because what we did is we tried to stratify the sampling of those lakes at a similar time of their growing season. And so this is what we started, our sort of COVID project, was we enlisted more than 100 participants worldwide in this DOM seasons network facilitated by the Global Lake Ecological Observatory Network, GLEON. And using a standardized protocol, we've collected DOM from these 70 sites worldwide every month during 2021. And hopefully we'll have some answers soon about how chemodiversity varies seasonally. And we've also recently discovered that chemodiversity influences how microbes use plastic-derived carbon. And that's a really exciting paper we have coming out soon that you can ask me about at the end of the talk. But again, like I really want to just emphasize that the take-home message here is that you should give a serious function about chemodiversity. Almost done. So I've told you about how chemodiversity arises, but I want to just finish off thinking about what controls. Sorry, I've told you about why chemodiversity matters, but now I just want to finish off thinking about how does chemodiversity even arise, right? What controls DOM, dissolved organic matter, composition, and persistence? And that remains actually quite contentious. And so what we've discovered in the lakes here, don't worry about this, I'll explain it in a moment, is that lake functioning and the chemodiversity gradient more broadly was driven by the turnover rather than the production of labile compounds. So each point here in these plots is one molecular formula from our lake survey. And along the y-axis here, we have the H to C index, which is the ratio of hydrogen to carbon atoms in the molecule. It's an indicator of increasing lability. And so along the x-axis, we've then taken the abundance of each molecule in each lake and correlated that with the net primary productivity of the catchment. So points further along the x-axis are molecules which are positively correlated with productive forest vegetation catchments. And then we've colored the points according to their correlation, to the correlation of each molecule with chemodiversity. And so the overall trend in this plot shows that formulas tend to be associated with more productive catchments are more labile, right? So there's a positive trend, more productive catchments, more labile molecules, and those more labile molecules are associated with less chemodiversity, lighter blue colors. We find similar trends in this plot here with what we have here is a correlation with microbial respiration. So formulas that are positively correlated with microbial respiration tend to be more labile and correlated with less chemodiversity. So the kind of take-home message here is that the productive catchments are producing more labile compounds that are more rapidly turned over by microbes. And that's what's generating, or that's what's actually in that case, reducing chemodiversity. And so the last little bit I'll talk about is an experiment, a forest harvest experiment. We're going back up into the forest now, and it's an experiment we've been conducting with the Canadian Forest Service outside of Sault Ste. Marie in northwestern Ontario to try and understand how dissolved organic matter composition and persistence varies across the landscape from soils, from the origins up in plants all the way down into lake headwaters. So as part of her PhD, Erica Freeman has been running this 3-year experiment, like I said, outside of Sault Ste. Marie in a place called the Turkey Lakes Experimental Watershed. And she has 4 replicated catchments, 2 of which were logged after 1 year. And she's been tracking the dissolved organic matter at 4 different depths at each of 5 different topographic positions in the catchment, again, at like every 2 weeks throughout the growing season for 2 years. And the main discovery here is that dissolved organic matter actually converges towards this pool of compounds that are found everywhere, a universal pool as it passes along these environmental gradients and is increasingly reworked by microbes. So this is the study design. And what we have here, like I said, we've got this hill slope where we have pore water samplers at different positions along the hill slope. And then we're sampling water. We have pore water samplers. We're pulling up water from 5, 15, 30, and 60 centimeter depths at each one of these positions. Keep going. Okay, and so it turns out that perhaps unsurprisingly, of the molecules that we find everywhere, these universal compounds, like 80% of them turn out to be lignins that are coming from plant material. And what Erica discovered was that as we get to the bottom of these soils, these universal compounds actually become more abundant. And so what we have here is the estimated change through that soil depth profile, averaged across all the different hill slope positions of the proportion of those universal compounds in the dissolved organic matter pool. And so each point here is just one separate pore water sampler in a catchment. And what you should see is that there's a slight increase as we get down to the bottom. This is like a 4% increase, which on a relative basis is actually quite large, because we go from about 20 to 25% of the molecules being these universal compounds. And it turns out that it wasn't because more of these universal compounds were being produced, because we actually looked at their abundance, and it wasn't because they were necessarily resistant to degradation. In fact, we found that the molecules in this dissolved organic matter were actually getting larger in mass, which is known to happen when you have small plant-derived molecules like sugars. They're consumed and transformed by microbes into these larger molecules. And so that's what we're just showing on here. We have a measure of the molecular mass of the dissolved organic matter pool, and it's increasing as we go down through the soil, and it's increasing as we move down this hill slope gradient. And we see larger changes when the molecules, when we actually decompose all of this into the individual compound classes. And I won't show it, but we've further attributed these changes in these compounds to the expression of important microbial genes, which turned out to be much more important in an analysis. We had like Erica suffered lots. She just loved torturing herself. She measured over 400 different variables that we co-measured with the dissolved organic matter, metatranscriptomes, metagenomes, metabolic activity like enzyme activity and physical chemistry. And out of all of these 400 variables, what turned out to be the most important were these genes controlling the expression of enzymes involved in organic matter degradation. Okay, so just to kind of wrap up, all this work, both from the soils and the European lakes, is very consistently telling us that the composition and persistence of dissolved organic matter is very much created by prolonged exposure to microbial reworking. And so one thing we're really interested in understanding now is, how do microbes even evolve to use different organic matter sources? How quickly and consistently can they adapt if these sources change because of climate change? And what is their contribution towards consuming versus creating chemical diversity? And this is my last slide, I promise. And so as part of that ERC project, we're actually running a long-term evolution experiment where we're tracking the genetic adaptation of bacterial communities grown on different environmental mixtures of organic matter for thousands of generations, to try and see how to try and answer those questions I just told you about. I think that's all I'll say about that. So, yeah, because we're sort of there on time, aren't we? Yes. Yeah. Okay. Gone over. Okay. So I hope that I've convinced you today that there's this really remarkable diversity of organic matter that is key to directing the function of lakes and the fate of carbon as it moves from land into the oceans, and that largely happens by influencing microbial activity, and that climate driven shifts in these terrestrial inputs, I would say, are important for like brewing different mixtures of organic matter. So it's kind of like, I don't know, do we have tea there in the back? But what you can do is you can soak the exact same amount of material, see, same amount of tea leaves in all these cups, but you get very different properties depending on the type of material you soak in it. And so a key challenge now is really taking this predictive understanding that we've tried to generate and upscaling it into meaningful ways, whether that be influencing forest management for drinking water or integrating microbial processes into these Earth system models of the global carbon cycle. So if you're interested in what I've talked about today, we're actually hiring at the moment 2 postdocs in my group at Cambridge to work on these projects, and we also have funding for master's and PhD students to start next academic year or I think the following one on areas that I've talked about at Trent University in Ontario. So if you don't want to go to the UK, master's or PhD, we're looking to carry on, come talk to me. We've got funding to work on these things.
Not yet.
So yeah, work that I presented today wouldn't be possible without countless postdocs, PhD students, technicians, and undergrads, and I really need to acknowledge everyone's contributions. Thanks for listening through all of that. I think I've gone well over, haven't I? We started late. Yeah. Thank you.
Thank you.
But I'm happy. Like I guess if people have to go, but we can. If there are questions, we can sit and talk. But if some people need to escape because I've talked lots, they can. I won't be offended if you just leave now.
Yeah.
Prof, thank you for the presentation. It was really interesting and really easy to understand for non-specialists. I just have like a kind of vocabulary question. So you went to the west for your peatland studies. Yeah. So you said you you studied like discontinuous permafrost peatlands and sporadic permafrost peatlands, and I don't really understand the difference.
So it just has to do with the amount of permafrost in the catchment. So continuous permafrost is just like all the land is got permafrost underneath it, and the sporadic or discontinuous zone, there's a smaller proportion of the soil actually has this frozen ground.
At the beginning, you show one of the slides showing how zooplankton, the 40% of the biomass of zooplankton, 40% is organic matter that come from the forest, allochthony.
Yeah.
But you show like different lakes in Sweden, in Canada, in United Kingdom, in Scotland, no, it's in Scotland. And my question is, did you show one different patterns in the percentage of biomass for zooplankton in the different lakes, or is what like the same, the lakes?
For the actual biomass,
Yes,
The allochthony.
the allochthony.
for the allochthony, yes. So the allochthony varied quite a lot. Do you want me to go?
Yes, but it was the same among the lake, or is among the different countries, among these things?
Space geographically,
Yes, yes.
no, it didn't.
Yes, I want to have this because I feel that it's impossible that this is the same. Too.
So there was, it went all the way from like
Mhm.
probably 8% to like 94%
Okay.
in terms of how much allochthony there was.
But among the different.
And it was, so it wasn't, yeah, so there would have been strong differences geographically
Yes,
across
what is the
these
difference?
lakes.
It was more in Sweden, it was more in Canada.
I don't remember actually, all, because what we did was we tried to link this to the catchment characteristics.
Because
So-
in one moment you say later that it's very interesting. Don't you say that the most important thing is to know what happened with the food webs, and we are going to go to the basics. And for me, I think the basis is not the food webs. For me, the basis is how much input of organic matter arrive. And the input, I think that could change the metabolism of the lake, could change food webs, could change everything. But especially this input depends on what? Of the thing that you finally say, natural disturbances or anthropic disturbances, like insect outbreak, forest harvest, fire, I don't know.
Yeah,
So
so
the thing is that in boreal forest, the boreal forest is harvest each day and each another time, and there is not so much places like trees with the lake. No.
So one of the things we did in this day, like I didn't show it because it's a super complicated path
Yes.
analysis. But for each one of the lakes, we mapped the catchments and we had layers of the forest cover, of soil carbon, of forest biomass.
Mhm.
And so these things were like the key predictors of allochthony.
Super nice.
So there was geographic variation, but we were interested in understanding what properties of the catchments actually would explain that geographic variation. So maybe like, yeah, in parts of Sweden where there is a lot of forest harvest, the allochthony might be lower because the forest biomass isn't there. The other thing that was really important was the physical connection to the catchment. So like I think it was the ratio of lake perimeter to lake area. So if you had a long lake perimeter and a big shoreline, small lake, so like a big skinny lake with lots of shoreline, then the effects were stronger allochthony.
I think that they study the impact of disturbances in the lake, in the food webs, in the lake, in the biological part, but also in the biotic part, what happened with the metabolism of carbon and everything. It's interesting because with climate change, the frequency and the severity of many disturbances will increase, and then it will be, we need to know what is the future, to understand this phenomenon right now. I have other questions. You can go to the slide where you show the insect outbreak, please.
Yeah.
Yes, thank you.
Where was that? Yeah, it was here.
Here is this good? I have one question about what is the insect outbreak that you were testing? It was
So these were defoliators. So
Yes, defoliator, but
bark beetles aren't common in Ontario. So mostly defoliators like gypsy moth and tent
Nice.
caterpillar.
Ten caterpillar.
Yeah, the two of them.
Do you know what is the main insect outbreak in Eastern Canadian forest?
The spruce butterworm.
Spruce butterworm,
Yeah,
exactly.
yeah.
And you didn't consider this in the study.
Spruce butterworm, I think, is in there. So we basically looked at, so the data, the disturbance data is the Canadian Forest Service do flyovers over this whole area, and they just map what's disturbed and the species. So we didn't really care so much about the species. We just classified things as defoliators, and we didn't look at bark beetles because there aren't many bark beetles, at least like in this central part of Ontario.
For mixed forests and deciduous forests, ten caterpillar is very good because it's the main disturbance there.
So the
Right.
supplementary materials of the paper, like I can't remember, but they have the amount of area that's affected by the different species.
In-
So we did know what the species are, but we did look in detail. We did look in detail at the species data, because it was mainly tent caterpillar and gypsy moth, like overwhelmingly, that's what it was in these sites.
In this kind of forest.
Yeah.
I mean,
Yeah, if you stay in the slide, I have one question because I didn't understand when you were speaking about, and you say that the poop of the caterpillar are considered like inorganic in your. So basically, you were seeing increase in inorganic carbon, but why the poo is considered like?
Yeah. So that's kind of a consequence of the water chemistry data we have. So they don't measure dissolved organic nitrogen. So we think that there's some breakdown of that poo that is increasing inorganic nitrogen forms as well. So if I've understood your question correctly, so the poo is organic,
Yeah.
but then why are we measuring dissolved inorganic carbon,
Yes, exactly.
nitrogen? Yeah, because that's what the data we have.
But because you link that data to the poo of the caterpillar.
Yeah, so the thinking is that as that's broken down, as the frass is broken down into soils by microbes, they're releasing like inorganic forms of nitrogen that then get washed into the water. Presumably there would be an effect of dissolved organic nitrogen as well, but we don't have the data.
Okay, just because I didn't understand.
No, the water chemistry data comes from like routine historical water chemistry monitoring by the provincial government and other collaborators. So this is like, we couldn't change anything. These are the 30 years of data we had, and that's what we had. But the thinking is that it would also be affecting inorganic and organic forms of nitrogen.
Yeah. And concerning the dissolved organic carbon, how the poo influence the dissolved organic carbon?
So what we think happens, right, is the main source is leaf litter.
Yeah.
Of dissolved organic, or a big source is leaf litter.
And why? Because why not the poo of the?
Well, what's happening is the caterpillars are eating the leaves, so the leaves aren't available to fall in the water to provide, or on the soil to provide a source of carbon. And what they're doing, because they're taking the carbon out of the leaves, they're assimilating that to grow.
Yeah.
And then they don't really do much. They're very inefficient assimilators of nitrogen. So that's why the poo that comes out is actually very skewed in terms of high like, or low C/N ratio. So lots of nitrogen, little carbon.
So basically, they respire a lot of carbon, but they do not export. A lot
Exactly, they're respiring it, or they're assimilating it, they're using it just to build biomass, and they're not exporting any of the carbon, they're just exporting nitrogen. So yeah, they just convert the leaves into nitrogen is what we think happens.
Okay. Another question is about I still don't understand the link between the negative correlation between MPP and chemiodiversity. You can explain again.
Yeah, so there what we think is happening is that where the forests are more productive, and like it's a much more complicated analysis I didn't get into, where the forests are more productive, the organic matter that they produce, for example, tends to be more enriched in nitrogen compounds. And so those compounds are much more labile.
Yeah, but
And so when they're labile, the compounds are degraded by microbes.
Yeah.
And so the chemodiversity decreases.
you know, because you don't measure what is labile, because the labile go away. You measure what remains.
Well, that's debatable, yes. Yeah. So that's like a criticism of, yeah, of dissolved organic matter. The way we sample it, at least the one-off time point, you're right, there isn't a time series, is that you're assuming, yeah, maybe what was labeled 24 hours ago isn't there, but what's labeled at the moment maybe you might still catch it.
Okay.
But I agree with that point.
Okay.
That's a fair criticism, that we're only measuring some of what's labeled, because what is labeled is by definition, if it's labeled, it's not there.
Yeah.
But that's changing the whole time, right? There's labeled compounds, like if you can think of it, it's almost like they're labeled compounds every day. So if we go on today, we might miss yesterday's labeled compounds, but maybe we'll catch the stuff that hasn't been consumed yet.
But I just want to know if in the analysis, for example, if there is a lot of diversity, but one compound is predominant, you get.
Yeah, so this is really interesting about like the concept. So this is one of my PhD students is thinking about like the idea of species abundance distributions, from like traditional ecology, and how that idea ports over to dissolved organic matter. So are there like some compounds that are super abundant and they're what dominate? And I don't have an answer for you. It's something that I think is very interesting to look at.
Just because the idea is that if one is very predominant, you catch only the one that is predominant, but you don't see the.
Oh, we are measuring everything, right? Like, so in one of these, so with the techniques, the instruments are such high resolution that yes, you can detect things at very low concentrations with these instruments, like incredibly low concentrations. So yes, that might be the case. Some molecule is more abundant, but the instruments have such high resolution that you will pick up everything.
Okay.
At least within the analytical window. So the other like caveat is that the mass spectrometer only measures a certain size fraction of the dissolved organic matter.
Sorry, I have a little question. Thank you for the presentation. Yeah, it's very interesting, especially the chemical diversity and also the dissolved organic matters separated, completed with all over the world. But I have a little question about greenhouse gases, because I've seen that you have measured greenhouse gases during your project of peatland lakes in the permafrost zone, and also the ecological effect evolutionary dissolved organic matters. I guess you have only. I guess you measure once a year because it's really hard to access in peatland and also
The greenhouse gases.
Yeah.
So that varies. Yeah. Sorry, maybe that's not your question.
I guess it's your frequency of the sampling would be once a year because you have a lot of lakes and also it's really hard to access in the permafrost zone.
So the permafrost study was actually sampled several times during the ice-free season. Yeah. Again, it's that accessibility of the road. You could just drive up and it's a 16-hour drive, but that's doable. It doesn't cost 2,500 to get on the plane. So that permafrost lake study actually is several times over the year. It should be.
For another one, for over the Europe, have you also?
That's just one off. Yeah.
So the question is that how to evaluate the seasonality for the greenhouse gases emission?
Yeah, that's a good question. next step.
Next step. Yeah. Yeah. Okay. Thank you very much, Andrew.
Well, so I don't know what we have planned now, but I'm happy to talk to people.
Yeah. So, but maybe you're a bit hungry.
Yeah, maybe we go outside.